All right, so now we're going to talk about differences in hormone synthesis between um, the, and we're just really going to talk about the difference between hormone synthesis in terms of the peptide and amino acid derived hormones versus the steroid hormones. So, the peptide derived hormones. So, nearly all peptide hormones are actually at, synthesized as first as inactive precursors. Okay? And then they are converted to the active hormones. These inactive precursors are often much, much longer in amino acid sequence, and the activation process involves the cleavage or the removal of excess amino acids. Okay? Now, the terms that we use to describe um, these precursors, sometimes we call them pre-pro or pro. Okay? So, an interesting example of one of these precursors or how this process works is a protein sequence that we refer to as the pro-opiomelanocortin protein, okay, or sequence, the pro-opiomelanocortin sequence. So this is interesting because this is a single. This is so there's one gene that codes for this sequence, okay, and the longest version of the sequence is called pre-pro opiomelanocortin. And after this long chain is produced, it then undergoes processing. It's something called post-translational modification, right? So after translation, after the protein is produced, it's modified. So it starts to get cut down, and as it gets cut down, it forms several active peptide hormones. Okay? And so some of these active peptide hormones include a hormone called melanocyte stimulating hormone, or MSH. Okay? And there's MSH gamma, there's MSH alpha, okay, MSH beta, and it's a hormone that stimulates melanocytes in the skin to produce melanin. Uh, it it also is cleaved to produce corticotropin, or ACTH, which we will learn about a little bit later. It produces a hormone called lipotropin. It produces endorphin and enkephalon. And endorphins and enkephalon are actually opioids, which is why that term opio is in the name. Okay? So one polypeptide sequence gets modified after it's produced to release multiple active hormones. Another example of this process is the production of insulin. Okay. So insulin is first produced as a long chain of polypeptides that we call pre-pro insulin. Okay. Then after that pre-pro insulin is translated, right after, and there's a gene that codes for the pre-pro insulin polypeptide sequence. So after that gene is translated and the protein is produced, it undergoes cleavage, and the first cleavage step produces a shorter polypeptide chain that we call proinsulin, and then it actually undergoes a second cleavage step that produces the active insulin. Okay. Now, this process, we're still on peptide hormone synthesis, and this process of producing a long pre-pro or pro inactive hormone, and then cutting it down to get an active hormone. Another example of this is a hormone called angiotensin II. So angiotensin II, this guy here, is actually the active hormone, but it's first produced in the form of what we call angiotensinogen. And there's two cleavage steps. to produce the intermediate angiotensin I, which is inactive, and angiotensin II, which is the active. Now, the interesting thing about this example is that unlike the previous two examples, this process of cleavage does not happen inside of the secretory cell. Okay? So the previous examples, the pro-opiomelanocortin protein getting pared down and the insulin, the pre-pro-insulin protein getting pared down, those two examples occurred inside of the secretory cell. Okay? So that the form of the hormone that gets ultimately released into the bloodstream is actually the final active form. In the case of angiotensin II, however, the liver produces this precursor. The liver produces angiotensinogen and releases it into the bloodstream. And then angiotensinogen, while circulating through the body, interacts with circulating renin to produce angiotensin II. And then angiotensin II enters particular tissue beds in the body, especially in the lungs, that's rich in what we call ACE, or angiotensin converting enzyme, and that produces angiotensin II. So this process of activating this hormone actually happens in circulation rather than inside of the secretory cell. And when we get to the renal system, we'll talk about angiotensin II in much more detail, and you'll see sort of why that's true, okay? Why there's an advantage to having this happen in the bloodstream rather than inside of a cell. Steroid hormone synthesis, I mentioned before, always is derived from cholesterol. Now, the process of producing a steroid hormone actually starts at the liver. It starts at the liver because the liver is responsible for creating packages of fats, of triglycerides, and of cholesterol that we refer to as LDLs, or low density lipoproteins. And now these LDLs, and you probably have only heard of LDL in terms of being bad, because it's when you get your cholesterol checked with your clinician, they'll tell you that there's a good cholesterol HDL and a bad cholesterol LDL. But the body needs LDLs. Okay, so the LDLs can be bad in high amounts, but the body needs LDLs because LDLs are how the body transports cholesterol throughout the body. Or it's how cholesterol is transported throughout the body. So LDLs are little packages where we've got a phospholipid outer core, and then inside we've got just loads and loads of cholesterol, a little bit of triglyceride. And it's a nice way to transport this lipid-soluble chemical, this lipid-soluble molecule called cholesterol. Okay. So these LDLs travel in the bloodstream and they get to a cell that produces a steroid hormone, for example. Okay. So this cell might be a cell in the adrenal cortex, and we'll learn about the adrenal cortex more in class. But LDL will bind to the LDL receptor on this cell. It'll be taken up through the process of endocytosis, and then the cell is going to start taking apart this LDL in order to liberate the cholesterol. And then from the cholesterol, the cholesterol interacts with various enzymes to produce intermediates and ultimately our whatever steroid hormone we want to produce. So the process of producing steroid hormones involves a series of biosynthesis reactions that requires the production of intermediates. So here's our buddy cholesterol at the top here, okay? And let's say we want to produce testosterone. Okay? So let's say the cell that's taking up cholesterol um, is a cell in the testes, okay? And it wants to produce testosterone. In order for this cell to produce testosterone, it must first produce pregnenolone, okay? And from there, so it has to have the enzyme necessary to convert cholesterol into pregnenolone. And then from there, either it can go across and produce OH pregnenolone, dehydroxyepiandrosterone, and so forth. Okay, so either it's going to go across and down to get testosterone, and it has to produce all those intermediates, or it can go from pregnenolone to progesterone, and from progesterone to dehydroxycorticosterone, and then go across. Okay, and which direction it goes in depends on which set of enzymes that particular cell happens to possess, right? So at each of these arrows, there's an enzyme necessary to make that step. Okay? But you can't go from cholesterol directly to testosterone. You have to go through these intermediates. The same is true, let's say, if you want to make aldosterone. If you're the adrenal cortex, you want to make aldosterone. You have to first make pregnenolone, right? So pregnenolone is the first intermediate. And then you have to produce corticosterone and go down, right? Or some variation and go across. So the production of steroid hormones requires the cell to possess the particular enzyme necessary, right, to get to the ultimate steroid hormone. And it also requires you to produce intermediate hormones.